Thank you all so much for joining today's webinar. I'm joined by my colleagues, Dr. Merit Sokovich and Dr. Sabrina Paganoni and Catherine Small on this webinar. And we are excited to present to you a new program that was recently launched called the Healey ALS MyMatch program. And this is launched by the MGH Healey Center. And this is an early phase clinical trial program. And I'd like to share with you about what ALS MyMatch is all about. Next slide. We are all aware of the ALS clock, and this is an ALS clock that we really want to beat. And it is a clock that ticks fast. And in order to beat this ALS clock, we need effective therapies. We need transformative therapies. In addition to transformative therapies, we also need efficient pathways for testing these experimental therapies and drugs. We want to have mechanisms by which we can match participants to the right trials based off of their disease biology and biomarkers. And more importantly, we want to make sure that there is rapid access to high impact trials for ALS individuals. Next slide, please. ALS is really in an exciting time. There's a large pipeline of investigational products. The current numbers exceed 100 investigational products that are either in or entering early phase clinical trials. And this includes phase 1 studies or phase 1B or 2A clinical trials. And around 34 investigational products are in late phase trials or phase 3 trials. Next slide, please. We already know about the Healey ALS platform trial and how it has really revolutionized clinical trial design and access and testing pathways in the later phases of drug development. There are a number of efficiencies that the Healey ALS platform trial has already put in place in terms of providing rapid access to large numbers of ALS individuals within short periods of time, but also having a mechanism to test multiple drugs at the same time with sharing placebo and so on. And next slide, please. What we want to do now is really think of the step before the Healey ALS platform trial. The Healey ALS platform trial is a late phase clinical trial that's primarily looking at clinical efficacy testing to understand whether a drug has a clinical benefit or not. But there is a big portion of the drug pipeline before the Healey ALS platform trial where a number of drugs are entering clinical trials, but there are a number of unmet needs that we have to tackle so that we create a seamless pipeline or an efficient pipeline through which drugs can then move forward from the early phase testing to the later phase testing. And in the early phase testing program, we want to really identify the right trials for the right people. And we know ALS is very heterogeneous and it's not one disease, so this one drug may not be beneficial for all ALS individuals. And how do we then identify subgroups of people who may benefit from certain classes of medications or certain therapeutic approaches more than others? And we want to really understand before we put drugs and therapies in clinical efficacy trials, whether these drugs or experimental compounds are doing what they're supposed to do. Are they reaching the brain? Are they reaching the motor neuron cells? Are they engaging with the target that they're supposed to engage with? Are they shaking hands with the protein they have to shake hands with? And we want to know all of these things before we move forward into large, several hundred participant clinical trials to understand clinical efficacy. We also want to know whether we're bringing forth the right dosage of an experimental drug into a clinical efficacy study. And we want to do this in a way that we want to learn all of these scientific questions early on before a clinical efficacy study. And we want to do this in a way that really improves and expands access for patients to clinical trials. And in a way that we can expand clinical site capacity that allows for more ALS individuals to participate in research and clinical trials at their respective sites, and also reduce the travel burden or outcomes burden. We know that a number of clinical trials may have several visits where you have to travel to a site and a long list of outcomes that you may have to complete. And how can we be mindful about all of these things and create options that are flexible but not lose rigor? We know that clinical trial drug development is also really expensive. It's becoming more and more expensive, and we want to make sure that for drug developers entering into the ALS space, we make clinical trials affordable and design trials trials that are affordable and bring high impact trials to the ALS space. We can go to the next slide. This is how we are positioning the ALS MyMatch trial in the drug development pipeline. So if you think about the the Healey platform trial as a late phase study, the Healey ALS MyMatch trial is a precursor to the Healey ALS platform trial. It is an early phase trial program where we understand all of those questions that we just talked about. And if they're successful, then the successful drugs can then move on to either become a regimen of the platform trial down the road, or could potentially become a standalone trial that could be run at multiple sites within the US or even internationally for that matter. So next slide, please. And this is a schema of the ALS MyMatch program. So the idea of matching the right trials to the right individuals is really about identifying what those disease biomarkers are, whether it is a specific gene or a genotype or a specific biomarker, blood biomarker level, or a type of blood biomarker or an imaging biomarker that could then identify whether somebody has a certain disease pathway that would be beneficial for that particular drug that we're testing. Whether we're testing, for example, if you're testing an anti-inflammatory medication, we want to make sure that people who are entering that study may have inflammatory signals so that we can get the maximum output from that trial. So in the ALS MyMatch program, we start with a group of heterogeneous ALS individuals who will then participate in a common screening protocol. The idea here is to run one screening visit and potentially use that screening blood samples and data that could then be evaluated for eligibility for multiple ALS MyMatch trials at the same time. And depending on what biomarkers or what trials may be more suitable, then potentially those are the matches for a particular individual. There could be more than one match, but an individual will have to then choose one trial that they want to participate in and move forward in that trial. Each of these trials are designed and implemented as independent clinical trials, and each of them will have their own set of outcomes, the duration, and the scientific questions that they're looking at in each of those trials. In addition to the drug development goal, we're also interested in understanding how to then identify the right population, and how do we then understand subgroups and the optimal population for clinical trials moving forward. And part of that is really understanding the genetic makeup of ALS individuals. So all individuals will undergo genetic testing, DNA banking for whole genome sequencing to understand these patterns. And another innovation in this program is to save stem cells from participants in the ALS MyMatch trial, which can then be used for testing in a laboratory setting on how the drug might have a benefit on motor neuron cell lines if a clinical trial is successful, and we'll be testing the stem cell lines of those participants in that successful trial to then have a second method of validating whether a drug has a benefit or not. So this is the overall schema of the ALS MyMatch program, and we really want we are positioning and designing this program to be very patient-centric, being mindful of all the places where we can create efficiencies and make it as easy and less cumbersome as possible for patients to participate in trials, and at the same time generate value-informed drug development that could potentially accelerate the speed of drug development for all of ALS by creating a pathway for the next phase of testing. Go to the next slide. So the scientific scope of ALS MyMatch is one step before understanding whether the drug has a clinical benefit or not. At this stage of drug development, we don't know if these drugs have a clinical benefit or not. At this stage of development, we want to understand whether the drug has target engagement and are we moving the right dose forward based off of the biological efficacy and safety and tolerability. All clinical trials will also be looking at safety and tolerability very closely. And ALS MyMatch is all trials will be looking at identifying the optimal population for future trials. You can go to the next slide. This is a general design of what an ALS MyMatch trial might look like. Not all trials will look exactly like the way that you're seeing on the screen right now, but it could be a slight different variation of the schema that you're seeing. Essentially, there's a screening visit and a baseline visit, depending on the trial and the design. Potentially, these could be combined into one visit rather than having two separate visits. And then there are a few in-person visits and interspersed with some phone calls to make sure, or virtual calls to make sure that safety oversight is maintained throughout the trial, because every participant's safety is our top priority in any trial that we conduct. And each of these, during each of the in-person visits, there may be a set of outcomes, whether it's blood draw or maybe some spinal taps, because we want to understand what those protein changes are or biomarker changes are in the spinal fluid that directly bathes the brain and the spinal cord, and we can get a lot of information in the spinal fluid that we may not be able to get in blood directly. And there are some questionnaires to fill out, and each of the clinical trials are designed to be either 3 to 6 months long, depending on what the scientific question is. And depending on the drug and the scientific question, these trials may or may not have a placebo in them. It really depends on what we're trying to answer in the study. And depending on the trial design, it may be an experimental drug that is novel to the ALS space, or it has never been FDA approved, or it could also be a repurposed drug, a medication that has already been FDA approved for a completely different indication, may perhaps even be a non-neurological condition that we're trying to understand if there could be a benefit in ALS. Or it could be gene-targeted approaches or cell-based approaches or immunotherapies or monoclonal antibodies, and a variety of different types of drug classes, or even perhaps devices for that matter. You can go to the next slide. And the buzzwords for ALS MyMatch is really about building efficiencies. It's about speed. It's about bringing access to high impact trials for participants. And it's really about not losing the scientific rigor along the way, and designing these trials in a way that we can get the answers that we want by also creating a number of operational efficiencies. For example, the sites that are selected for the ALS MyMatch, we're going to start with 4 sites, and each of these 4 sites are well-seasoned, well-experienced centers, ALS centers that are known as accelerated centers for enrollment. These are sites that are generally high enrolling, high volume sites. Again, we want to run faster trials to get those faster answers. And we will be using a single IRB that will govern the clinical trial across all the participating sites. And ALS MyMatch trials are all NEALS affiliated clinical trials that brings in also the resources and infrastructure and network of the NEALS participating sites and the subgroups and the patient community from patient advisory community from the NEALS that will also be advising the ALS MyMatch program. And each of these trials are really thoughtfully designed to think of the minimum required visits for participants. What are the minimum required outcomes that we need to collect? And there are also a number of options that are built into the trials where we can run local safety labs, as opposed to needing to have the participant travel to the site every single time for a blood draw that needs to be sent to a central lab. And using templated protocols and trial documents also help a lot. And our idea here is really to save on time. We really want to shorten the trial startup timelines. And these are important trials, and we want to bring efficiency in a way that we have the most efficient way of starting up a clinical trial, but also a faster access to a new trial in terms of the first patient, first visit, and overall enrollment timelines to really accelerate the number of participants who can get access to a trial at any given site at any given point in time. And our goal is via the common screening protocol to really shorten the referral to trial wait times. There are a number of options built in for remote consent and possibility of potentially maybe even combining a trial common screening protocol screening visit with a clinic visit, as opposed to requiring a separate visit altogether, depending on the site and the way the studies are run at that particular site. Go to the next slide. While we do all of this, there are a number of guardrails and checks and balances that ensure that each of these studies are designed and conducted with the highest level of rigor and patient centricity. We are assembling a patient advisory committee for the ALS MyMatch program, and that will help us sort of steer the decisions in terms of patient-centric trial designs and operations. There is a trial design and implementation leadership team. There's a therapy evaluation committee that vets all the incoming applications. We want to make sure that we're putting the right drugs that have impact and are a good fit for and relevant for ALS that are entering into the MyMatch program. And we have a separate biomarker expert advisory panel. All of our trials are biomarker-driven, and biomarkers are central to the program. We have an expert panel that guides us on what the most optimal biomarkers would be for any particular trial, as well as at a program level overall. Go to the next slide. And this is our therapy evaluation committee who are helping us vet all the applications that come in for being included in the ALS MyMatch program. And we're very thankful to everybody that is contributing to this committee. You can go to the next slide. And this is the Biomarker Scientific Advisory Expert Panel. And this is a core panel that who will help us with priority setting, as well as identifying ALS disease biomarkers and designing some of the biomarker portions. Several expert cores in very specific areas, for example, cell-based assays or stem cell experts or imaging experts, depending on what the trial might need, or even thinking about multi-omics, genomics, proteomics, or metabolomics approaches for the trial scientific scope as may be required for each particular trial. Go to the next slide. And the most important thing is, as I mentioned before, patients are at the front and center of everything that we do in ALS MyMatch. And our goal is, while we are developing therapies and drugs for all of the ALS community, we want to make sure that participants who are participating in these trials have access to high impact trial opportunities. And that's really important. And while we bring these access opportunities to trials, we want to make this experience as efficient and less burdensome as possible for our ALS participants, reduce the trial fatigue, and engage as early as possible. And our goal is to reduce those wait times after diagnosis, after a referral to the ALS MyMatch program, and try to build in ways to then try to combine the study visits with clinic visits wherever possible. Next slide, please. So how can one become a MyMatch trial participant? There are 3 steps to this. The first step is, if you're interested in ALS MyMatch, the first step is to talk to your ALS doctor or our MGH research access nurse. And at the moment, most of the Healey ALS MyMatch trials are focusing on people with early ALS who have less than 24 months, from weakness onset, and who have a breathing function of more than 65%, and who have undergone ALS genetic testing and are diagnosed with ALS. But in the future, depending on the trial, these criteria could change. And once you are found to be eligible, if you meet these initial criteria, the next step, the entry point to ALS MyMatch, is the common screening protocol. And that will allow us to look at preliminary eligibility for an individual for multiple ALS MyMatch trials. If there are more than one trial if there's more than one trial that is running at any given point in time. And finally, the third step is to identify the right trial match and enter into the trial. And once you enter into the trial, you're sort of committing to that trial, to participate in that one trial. Go to the next slide. And our very first trial that we are launching in ALS MyMatch is called the Acacia trial. And we have an upcoming webinar on Monday. This is a Neil's webinar. Please feel free to take a picture of the QR code to register. And we're excited to share with you about the Acacia trial, which is testing a repurposed drug called digoxin in ALS. You can go to the next slide. And one more slide. I'd like to thank the leadership at the Healey ALS Center, Dr. Sokovich, for leadership and support, and all our colleagues at the Healey Center for making the ALS MyMatch program possible. And a big shout out to Niels for being a partner in the ALS MyMatch program, and our ACE centers, Northwestern University, University of Minnesota, Nova, Southeastern Florida, and Mass General Hospital as our ACE site participating sites in the ALS MyMatch program, and all our other funding supporters, including private foundations and individual donors who have made this program possible, and our colleagues and who are helping us through multiple colleagues outside of Mass General, including the Barrow Neurological Institute and others who are helping us bring this program to fruition. Thank you so much. With that, I'm happy to answer some of the questions.
Thank you so much, Dr. Babu. This was a fantastic overview. I also want to highlight that Catherine has kindly posted a number of links in the chat in terms of, tonight's webinar. This will be recorded and posted online if you want to watch it again, if you are interested. And also there's also the link to register for the Niels webinar on Monday. There's a lot of questions in the chat, and so thank you so much for sending them. Really, that speaks to the interest of the enthusiasm for this. So few things. So one question you just answered: what are the 4 locations again of the MyMatch, and is this for people eligible for clinical trials or for EAPs or both?
That's a great question. So 4 sites that we're starting the program with are our Mass General Hospital, Nova Southeastern in Florida, Northwestern University in Chicago, Illinois, as well as University of Minnesota. But this is just a starting point, and our hope is that as we bring in more experimental drugs and trials into the program, the number of sites will also expand down the road. And your second question, Dr.
Is it for people who are eligible for clinical trials or for EAPs or both?
Yeah, that's an excellent question. So MyMatch is an early phase clinical trial program, and we're focusing on people who are early in the disease at the moment. So this would be for people who are eligible for clinical trials.
So again, the follow-up question to that: what if somebody has had the disease for at least 2 years or 24 months? What are other options that are available to them?
That's an excellent question. As a community, we struggle with finding the right amount of resources and trials and research opportunities for all segments of the ALS population. We'd love to be there where we can offer a trial or an expanded access program for every individual who's diagnosed with ALS, but we want to get there, but we're not quite there yet. At the moment, this program, as well as a number of other programs, are really sort of looking at people who are within a certain time frame, 18 months, 24 months, or 36 months from symptom onset. But outside of that, there are expanded access protocols that are also ramping up, and there are opportunities through both NIH-funded expanded access protocols as well as other smaller programs as well. And there are off-labels as well that some individuals with ALS could potentially leverage.
Yeah, so there's a couple of related questions about repurposing drugs. So one question is, are you gonna include, new drugs or drugs that are already approved for other indications so that you can test them for ALS? And there is a specific question about a drug that was developed in the past for a different disease called SMA. And so the question is that something maybe that could be potentially could we repurpose that for ALS?
That is an excellent question. So MyMatch will include a combination of repurposed medications that have been approved for other indications, as well as novel experimental drugs that are being developed for ALS alone. And the decision of what goes into MyMatch and what doesn't go into MyMatch is really dependent on how strong is the science that's backing that particular drug, and what have you learned from preclinical studies or even other neurological diseases that would make us want to consider this for ALS? Is it relevant for the ALS disease biology in some ways? And all of this would be if there is an investigator, it could be an academic investigator, a clinician from a certain institution who's interested in developing a repurposed drug, has done a lot of work in their lab, or it could be a company who's interested in developing a repurposed drug. They would put in an application for ALS MyMatch, and that application will be vetted by the Therapy Evaluation Committee, which is really a committee of experts in the field of ALS who know the science and the biology of ALS. And once it goes through the vetting process, if it is deemed to be of potential relevance and fit and impact in the ALS field, then will be brought into the ALS MyMatch program.
Yeah. Is enrollment offered to international participants?
So we're not restricting participants to only people who are living in the U.S. or who are or U.S. citizens. Having said that, it depends on the trial and what the expectations are. Any trial is a two-way street. We are, as clinical trialists, our hope is to bring access to an experimental drug to ALS participants. At the same time, our mission is to develop therapies that would benefit a large, if not all, members of the ALS community. And so we want to keep that in mind. And in order to do that well and to do that with scientific rigor, there are a number of visits and procedures that we have to complete. So if somebody's living so can travel for any of the visits or complete procedures, it becomes logistically difficult in some ways. And depending, the best next step would be to really sort of talk to your ALS neurologist and clinician to see whether this would be a good fit for you as a patient to participate in any of the trials as and when we launch it. We have a dedicated ALS MyMatch website, that webpage that I think Catherine might have posted a picture. It was also on one of the slides. We can post that again. We will be updating that webpage as and when we have new information about new trials and opportunities. So feel free to check in periodically.
Great. A few questions about how you're selecting the drug. So, for example, can you tell us more if AI was involved in selecting digoxin, or what are the other criteria? Again, I know you mentioned that briefly.
We are living in the era of AI. There is no meeting and no webinar where AI is not mentioned. So AI is going to be a part of ALS MyMatch in a variety of different ways. For example, if there is a clinical trial where we're running an open label study of a repurposed drug, potentially we might consider designing the study to use AI-driven virtual control as a group to match to the participants in the trial to then analyze the data. And there could be drugs that are being selected for ALS MyMatch that may have gone through AI-driven approaches for drug screens and other things as well. So there are several different ways that we'll be using AI in the program.
Great. So speaking again about emerging technologies, will iPSCs, which are a form of stem cells, be used to match patients to treatments?
That is a really interesting and exciting question in many ways. So we're taking maybe one step prior to that. So what we're doing here is we want to kind of get to a place where we are able to match a trial based off of stem cells and the motor neurons that are derived from stem cells in a dish. But the first step as a field for us to understand is, can we then compare how people are doing in clinical trials, and can we compare how their stem cells or stem cell-derived motor neurons are doing in a dish? If the trial is successful, and then we look at people who respond to the drug and people who don't respond to the drug clinically and compare it to their stem cells in a dish. And so that's sort of like the first step of understanding how iPSCs could be used down the road in other trials, and that's exactly what we're planning to do in this.
Speaking of another cutting edge technology you're going to be using, you said everyone will have their DNA sequenced. There will be whole genome sequencing. So tell us more about it. Is it expensive? Are the results returned? How long does it take?
Yeah, whole genome sequencing is expensive, but there is no cost to the participant. That is important. And so any participant who is entering the ALS MyMatch will give a blood sample. DNA will be extracted from the blood sample and will be banked. And at the end of the trial, or even beyond the trial completion, to understand and to further the science of related to ALS and pathways and targets, we'll be doing whole genome sequencing. Unfortunately, we don't have the ability to return all of these results to participants because all of this is still sort of in the research realm, and we don't have definitive answers on what it means to find a certain genetic marker until it is actually fully established in the field.
Yes, so there's a couple of questions about people who are slow progressors. So for example, if somebody's a slow progressor, has had the disease, for let's say more than 2 years or even many more years, what are the options for research? Have you thought about including them in MyMatch, or are there other options?
Yeah, thank you for this question. That goes back to another comment about what are the options for people who don't qualify for some of the clinical trials. All of us, Dr. Paganoni, probably you can speak to this as well. We're all constantly thinking of how can we make access to experimental therapies feasible and a reality for ALS individuals. And both Dr. Paganoni and I and several colleagues at the Healey Center have led expanded access and are leading expanded access programs of all sizes, from a single participant expanded access protocol to large multi-site NIH-funded expanded access protocols. And this is really we're at the beginning of that era of really expanding the opportunities for non-clinical trial eligible participants. And we hope that we see more movement in that direction, and more companies and even federal agencies and NIH would continue to support and expand the support for expanded access so everybody can get access to these drugs.
Yeah, there's another comment about expanded access. First of all, I want to thank the person who wrote in the chat and commenting about participation in previous regimens of the platform trial and also participation in expanded access. So thank you so much for participating in so many trials and participating to expanded access. It's really amazing to see your contributions, and really we couldn't be making any progress without you. But so again, thank you for your participation. And then the comment is more about, some people have been on expanded access, and so can that lead to approval, or what's the next step? I guess when we do clinical trials and then expanded access, what's the process for going from testing to approval? Maybe if you can tell us more about that, understanding that different people can have, can respond differently to different drugs.
No, you're absolutely right. So any given trial, there are people, there's one section or segment of the trial population that responds, and there are others who don't respond. That's true for even the medications that have been approved. That are FDA approved for ALS and other indications. So when a drug does go for FDA approval, they take the entire data in totality. They look at the overall cohort and how did people who were treated with the active drug do in comparison to placebo. But in addition to that, there are also several subgroups that will be analyzed for what we call as responder analyses to understand what type of people are responding to this. Maybe there are people who have very similar overlapping disease characteristics, and maybe they have very similar disease biology. That's why they're responding to this. But those are all responder analyses that those in themselves will not lead to an FDA approval, but they can inform to identify subgroups of that optimal population that we talked about earlier in my talk, that potentially could then lead to another trial design that would then enrich just people with those exact characteristics to see if there is a benefit. And part of what we're doing as part of MyMatch is also to identify that optimal population that could then be used for future clinical trial testing as well.
I'll take a quick question. I know there's a question about a couple of the previous platform trial regimens, the one testing pridopidine and the one testing CNMAU8. So both have completed the platform trial and both are currently available as an expanded access option. And then the question is, when are the phase 3 trials going to happen? And so I know that the companies are actually getting ready to launch them. It's still in the startup phase, so I do not know when the companies plan on actually starting enrollment for the phase 3, but I know that's in the planning stages. A couple more questions for you, Dr. Babu. So some of the drugs that have been identified as promising, drugs for MyMatch may also be available for other indications. Again, that's the point of a repurposed drug. So what are the pathways for access, I guess? Is it more like off-label? What are the pros and cons of sort of off-label prescriptions versus clinical trial participation?
That's an excellent question. And, this is a big topic. It's a loaded topic. And how do we think about repurposed drugs? And what's the benefit of being in a trial where there is an opportunity to get access to a repurposed drug outside of a trial? And can you get access to a repurposed drug outside of the trial? So I think a lot of that depends on one, whether that particular drug is the right option for you as a patient. The first step is to really talk to your clinician, your neurologist, to see if that's the right option to move forward. The second is within the boundaries and realms and parameters of a clinical trial, there are lots of built-in checks and balances and guardrails and safety oversight mechanisms that are built in. Even if a drug is FDA approved for a different indication, there could easily be possibilities where it is not safe or it is not appropriate for ALS individuals. And we want to make sure that if there is an emerging signal, we catch it early. And because we are looking at multiple individuals who are taking the same medication with the same dosage and standardized conditions when the trial, it helps us to keep an oversight and maintain that oversight of every single participant very, very closely and with the highest standards possible. That sometimes if it's an off-label, you may not have the opportunities for the same level of, for example, for digoxin, cardiology oversight and the safety review committee that we're looking at every individual and the group's overall trends and things like that. So there are advantages to participating in a clinical trial, even for repurposed drugs, even if you have the option of getting that outside of a trial. More importantly, you're contributing to something that's, bigger, much bigger than oneself in terms of moving the science forward. If this is truly beneficial, you're contributing to a drug that potentially could then move forward to a larger study. We don't know that yet, and we're trying to understand that. And there are a number of questions we're trying to understand, but this is all part of that drug development program overall.
Yeah. It's a good question. I mean, I see that many people on the line have participated in multiple studies. So for example, let's say that somebody has participated to all ALS and already donated as part of other biomarker studies, such as all ALS, different biomarker samples, so DNA or spinal fluid or blood. Could those biosamples be used to augment the results of MyMatch, or are they studies separate?
Yeah, that's a great question. In the future, in our wish list, then it would be wonderful to then sort of harmonize and combine all these different studies. But the way the MyMatch program is developed is that all of the other programs are separate because there are there are parameters in terms of consent, data sharing, data use, and how are we using those samples. And there is also a window that we want to kind of check these biomarkers before trial entry. So a number of those factors would make it really difficult to pull results from a different study and to determine eligibility.
Yeah. So again, there's lots of great questions and lots of important information that Catherine put in the chat. Thank you so much. So I do want to, I think we took all the questions tonight, but I do want to highlight that there's going to be another webinar about MyMatch and specifically the digoxin trial. Is that Monday? Did you say Monday?
It's Monday.
Fantastic. And if everything will be recorded anyway and posted. But so with that, maybe I'll turn it over to you, Dr. Babu, for the final word before we close.
Thank you all so much for all these excellent questions. And these are all very relevant and important questions. And we appreciate, I can sense the energy and the enthusiasm for our program. And we're very grateful for everything that you do for the ALS community and participate in research. And we look forward to working with you in ALS MyMatch. Thank you so much.
And actually, I'm sorry, there's a question that just came in right now, so I do want to take it if you don't mind. So it's an important question. People who previously participated in different trials. So for example, specifically, the question is, if somebody participated in the Healey platform trial, are participants asked to participate in this? So I just want to start maybe answering, and then Dr. Babu, you can expand. But, there are, sort of, privacy rules, so we cannot basically reach out cold turkey to participants, but absolutely, if somebody participated in a previous study, such as the platform trial, absolutely. So first of all, thank you for participating. If you are interested in participating in another study, because for example, that particular trial was completed, finished, I would highly encourage you to connect with the same site, the same, coordinators or physicians or nurses that you worked with for referral. So we cannot sort of from our site go out and sort of reach participants, but your physicians, your team can advocate and send us, referrals to us. So I just wanted to share that again. We cannot sort of automatically invite people to participate, but you can either reach out to us, and obviously Catherine is on the call and is our patient navigator and available to make the connection, or your team, the people that you're working with at your clinic, can also send us referrals for this program or other programs. Dr. Babu, is that correct, or anything else?
That's perfect. That is perfect. Thank you.
Great. So with that, I think we'll adjourn the meeting, and very excited that you'll have another opportunity to connect with Dr. Babu on Monday if you have other questions. Thank you so much.
Thank you, everyone.
Thank you.